Hello everybody, I'm Noel Malod-Dognin. I'm a researcher at the Barcelona Supercomputing Center in the research group of Professor Natasha Prugel, and today I'm going to present you our work on chromatin network markers of leukemia. As you may know, chronic lymphocytic leukemia, or CLL, is the most common leukemia in adults. For a healthy individual, in the bone marrow, the stem cells will differentiate to create the different blood cell types, which include B lymphocytes. However, in CLL, there is a production of abnormal lymphocytes that are called leukemia cells. And when those leukemia cells take over the LCB lymphocyte, then the patients become subject to infection, anemia, and easy bleeding. CLL, in the literature, has been extensively studied from the sequence point of view. And for instance, whole exome sequencing revealed that there is a high degree of genetic variability in terms of mutated genes across the CLL patients. So from those sequence analyses, we also know 38 leukemia driver genes, and we also know that there are 5 common chromosomal alterations, which include 4 deletions and 1 trisomy. But what we want in our study is to go beyond sequence analysis, and we are going to do so by also using a new type of biological information, which is chromatin structure. So, as you know, the genetic information is stored on DNA molecules. However, these molecules are not simply floating around in the nucleus. The DNA, in fact, binds to several molecules, forming so-called chromatin, and this chromatin folds and acquires a very specific 3D architecture which is known to relate to the regulation and expression of genes. The key building block of that 3D architecture is chromatin loop. So what happens is that 2 regions that are far away along the DNA come into proximity in 3D space and are glued together with cohesin molecules. So here we want to capture that 3D architecture in a network that we call chromatin structure network, in which the nodes represent DNA elements, genes and non-coding RNAs, and where 2 nodes are connected by edges if the corresponding DNA elements are in contact in the chromatin. Since our chromatin structure are represented by networks, we are going to analyze them with state-of-the-art topological features that are widely used in network biology. And these tools are graphlets. Graphlets are small induced subgraphs that are used to quantify the wiring patterns around the nodes in a network. It's illustrated here on the right, where you can see the 2 to 4 node graphlets. And for any node in a network, we are going to create its graphlet degree vector, which is a vector in which each entry counts the number of times the node can be seen in any of the specific positions in the graphlets. So these graphlet degree vectors are used to compare the wiring patterns of 2 nodes in a network, but also at a global scale to compare the wiring patterns of 2 networks. So for our study, we collected the chromatin structure of 17 different human blood cell types. They are coming from the study of Javier et al. From 2016, which notably include the naive B cells, which will be our control cell type and whose statistics are here in blue. And we also collected the chromatin structure of chronic lymphocytic leukemia cells from the study of Beekman et al. In 2018. Whose statistics are represented in pink here. And importantly, all those chromatin structures have been captured using the same experimental methods in the same laboratory and in fact by the same group. So the first question that we asked is if the topology or the wiring patterns of those chromatin structure network relate to the functioning of the corresponding cells. So the first thing to know is that those 17 cell types, they're in fact divided into hematopoietic origins. So about half of them are lymphoid cells, including the naive B cells, and the other half correspond to myeloid cells. So what we did is we measured the distance between any two chromatin structure networks using the graph-led correlation distance, and we used those pairwise distances to plot the networks in 3D space. And as you can see, all the chromatin structure network corresponding to lymphoid cells are grouped together. All the chromatin structure network corresponding to myeloid cells are grouped together. And interestingly, the chromatin structure network of leukemia cells appear as a kind of outlier very far away from all the other blood cell types. So what we see here is that the wiring patterns of the chromatin structure network capture the phenotypic information about the cells. So the second question that we ask is, what can happen to the chromatin structure network of leukemia cell so that it appears as an outlier from all the other blood cell types? So the first thing is that we do was to compare the overlaps in terms of node and edges between the CS network of naive B cells in blue and of leukemia cells in red. As you can see on the Venn diagrams, the two networks globally have the same set of nodes with more than 70% of shared nodes between the two networks. However, in between naive B cells and leukemia cells, there is a strong rewiring since the two networks only share about 30% of edges. To visually see what could be happening, we then decided to embed in 2 dimensions the largest connected component of the two networks using so-called spring embedding, so that two nodes that are strongly connected to each other are in proximity in that 2D space. As you can see on the left, for the largest connected component in the networks of naive B cells, the nodes in blue tend to form well-separated clusters, and we can also see that there is a kind of hierarchical organization in the network. However, that organization seems to be lost in the chromatin structure network of leukemia cells on the right, where we cannot visually see these clusters of genes. To quantify this phenomenon, we use Louvain community detection algorithm to measure the number of communities in the different networks. As you can see on the plot, for all the LC blood cell types, we have a very similar number of communities. However, when we look at the chromatin structure network of leukemia cell, we can see that it has about 3 times less communities than the LC naive B cells. So whatever is happening in leukemia, it's inducing a reduced modularity in the chromatin structure network. So I'm not going to present this in the presentation, but it's reported in the paper. But in our study, we highlight the potential role of the cancer driver genes in this reduced modularity in leukemia. Then, after looking at the chromatin structure network from a global perspective, we wanted to have a closer look at the genes, and we asked if the wiring patterns around the nodes in the network relate to their biological functions. For this, we did 2 types of clustering and enrichment analyses. In the first one, we used spectral clustering to group together the nodes that are densely connected to each other. As you can see on the plot on the left, all the corresponding clusterings are significantly enriched in reactome reaction annotations. And we have very similar result when using reactome pathways and gene ontology biological process annotations. On the right, we clustered together the genes using their graph-led degree vector similarity, so that genes that have similar wiring patterns, but independent of them being in the same neighborhood, are grouped together. Again, we obtain very strong enrichment in terms of biological annotations, but interestingly, for the chromatin structure network of leukemia cells, the clusters that we are obtaining using spectral clustering are significantly less enriched than the one of the LC blood cells. And we do not observe such a decrease when looking at the graph-led-based clustering. This means that the rewiring that is happening in leukemia is altering the functional coherence in the neighborhood of the genes, but not in the shape of the wiring patterns. So since we have shown that the wiring patterns of nodes in the chromatin structure network relate to their functioning, we wanted to have a closer look at the cancer driver genes. So first we consider the chromatin structure network of leukemia cells, and the first things that we wanted to see was whether the cancer driver genes tend to have similar wiring patterns in that network. So on the left we have the two distributions. In pink, the distribution of Grafflet degree similarity between any two driver genes, and on the right, and in blue, sorry, the pairwise distances between any two background DNA elements. So as you can see, the driver genes tend to be more similar to each other than the rest of the nodes in the network. These mean that they tend to share a common topological signature. So to see what this topological signature could be, we plotted on the right the average Grafflet degree vectors of the driver genes in red and of the background DNA element in blue. And the black dots here represent the orbits for which there was a significant difference between drivers and background element. So as you can see, it's almost significant everywhere, which means that the cancer driver genes have a very characteristic and significant Grafflet degree vector signature in the network. So then what we wanted to see is if we can use that signature to uncover new leukemia-related genes. So to do so, we considered all the genes that are not known to have a role in leukemia and to prioritize them according to their Grafflet degree vector similarity to the known leukemia driver genes. In the table here, we present the top 20 prioritized genes for which we were able to validate that all of them have a role in cancer. So we validated this using either literature curation, or we also check if those genes are significantly differentially expressed in leukemia, or if there are significant prognostic markers of different types of cancer. So this result shows that in the chromatin structure network of leukemia cells, there exist structural hotspots in which the cancer-related genes are located. We also did the same to prioritize non-coding RNAs, and similarly we were able to validate that many of those non-coding RNAs are also related to cancer. But then we asked something maybe a bit crazy. So can we also find this structural hotspot in the chromatin structure network of healthy naive B cells? So this is what is plotted here. So in the chromatin structure network of naive B cells, you can see that the cancer driver genes also tend to be more similar to each other than the background DNA element. And they also tend to have a Grafflet degree vector signature that is significantly different from the background. And very interestingly is that we can also use this Grafflet degree vector signature to prioritize genes that are also cancer related. So here, what is very important is that we can predict these new cancer genes solely from the chromatin structure network of healthy cells. We also did the same for the non-coding RNAs, achieving comparable performances. So the conclusions. So in this study, we investigated the changes in cancer on the chromatin structure. We observed that the cancer-induced genomic rearrangement reduces the modularity of the chromatin structure network. We show that these changes involve a decrease in the functional coherence of the chromatin structure network, and especially in the node's neighborhood. Importantly, we characterize the topological signature of cancer driver genes, and this in both the chromatin structure network of cancer, but also of control cells. And we show that these signatures allows for prioritizing new cancer-related genes. Finally, our study demonstrates the existence of a structural hotspot of cancers in the chromatin structure of cancer and control cells. So from here, a key question that remains to be solved is how can these specific structures can help the mutations and dysregulation of genes. So I would like to thank you all for listening to that presentation. I would also like to thank all our team members, which include the members at the Barcelona Supercomputing Center in Spain, as well as the members from University College London. I would like to thank all the fundings that made this study possible. And finally, a little advert. We are actively looking for postdocs, PhD students, and research engineers. So if you're interested, then you can actually apply using the following web link. Thanks again.